Thank you for your very kind introduction. It's really a pleasure to be here. I am giving this presentation on behalf of myself and also David Thacker, who I worked with my online cannabis platform company, Tomori Pharmacology, doing business as Canify, and Michael Tagan, who is my business partner in consulting. These are our disclosures, mainly from Michael Tagan. And before I start the real presentation, I want to familiarize you a bit with some pharmacology terminology, because I'm of course very used to this, but most of you are chemists. And pharmacology is the study of drugs. I am a clinical pharmacologist, and it means that I study drugs in humans. And a very important part of clinical pharmacology is pharmacokinetics, or PK. And this describes what the body does to a compound. And maybe this picture looks a bit familiar to you. Pharmacokinetics is described by several phases, and you see a few of them here. So first, absorption. That's an important one to remember. Absorption means that the compound is absorbed into the blood circulation, the central circulation. So it goes into the blood, after which it is distributed. That's the distribution phase. I have no pointer, I'm sorry. And then it's distributed to different organs, for example, and at some point it will get metabolized, that means broken down. And it's broken down because it then gets more hydrophilic, so it can be excreted from the body. That's the lower side of the picture. So this is roughly pharmacokinetics, so that you get a bit familiarized with this. And then the other important aspect of pharmacology is pharmacodynamics. And pharmacodynamics describes what a compound does to the body. So now we're talking about effects. And we can talk about many different aspects of effects. And one of those is very familiar to you. In the previous session, this already was discussed. That is when you have receptors, compound bind to those receptors, and then a whole cascade of processes happen that will eventually lead to, potentially, an effect. And here I've got a very schematic picture of receptors. Those are proteins in the body that are binding places where compounds, ligands can bind to, and then an effect can occur. And here there are three different examples of receptors, just as an illustration. You have a full agonist binding on a receptor. That means that 100% of the effect takes place. You've got in the middle a partial agonist. That means that less than 100% of the effect will occur. And an antagonist, as opposed to what many of you think, antagonism doesn't mean the opposite of agonism. It just means that a compound goes to the receptor and it blocks the receptor, nothing can happen. So, in fact, nothing happens. That's an antagonist. Now, I want to do a little quiz because we already heard about CB1 receptor, cannabinoid type 1 receptor, and THC that binds to that receptor. So, there are three options, three possible answers. Is THC at the CB1 receptor a full agonist, a partial agonist, or an antagonist? I want to see the hands who thinks that THC is at the CB1 receptor a full agonist. I see and who thinks it's a partial agonist? And it's really funny how it is divided into parts. So now expect the middle part to, no, raise the hand. Who thinks it's an antagonist at the receptor? Yeah. So actually all of you are right. It's a partial agonist. So answer B is the correct one. And indeed, depending on which other compounds are around, it can act in an agonistic or antagonistic way. So partial agonist is the right answer. Okay, this was the introduction. And now we're going to the real part of the presentation, because we are here. Every conference has its own highlights, and here it's mainly about products. And many of you are either working at a product company or you are in some way related to a product company because you're doing work for them. And what happened over the past time is that more and more product companies arose. And you can think, well, that's very logical, because there are more states that legalize cannabis, so there are more product companies. But in my home state of Colorado, I sound really Coloradan, I know, Yeah, you see that after it got legalized in January 2014, both medicinal and recreational cannabis, there is still a very big increase of product manufacturers. It's still going on. I've not put the points in the middle, but believe me, it's still increasing. And you think, okay, that's interesting. That even in that state, there's still a development. What's going on there? Well, maybe more people are using more. That's very well possible. But if we look at these numbers of the number of dispensaries, that is increasing too, but by more than 2 times, whereas the number of product companies in the same period increased by more than 3 times. So this implies that per dispensary, there are more products and there are also more product brands that you can find. So a consumer comes in and has way more options. That's good for them, but is it good for you? Because if you want to stay afloat as a product company, if you want to get somewhere and be the one who is out there for the long term, you really need to distinguish yourself. And I think that one very important factor of how to distinguish yourself is by doing that with science. Now I want your participation again, because science-focused product companies, that is really a very interesting field. And if you're at the Emerald Conference, you are, I think, by definition, interested in science. But maybe I'm very naive. So I would like to see hands if you work for a product company. How many of you work for a product company? And I want enthusiastic hands, not like, yeah, okay, good. And keep your hands up, keep your hands up. I want you to keep your hand up if your company employs chemists, like what you have here, analytical chemists. A few go down, but many stay up, that's good. Now I want you to keep your hand up, because I want to know who of your companies employs a clinical pharmacologist? 1. Okay, I want to talk to you. No, that's very rare, and that's very interesting. Pharmacology, the study of drugs, you're making products to get particular effects, but you're not actually having someone on board who is specialized in how drugs work in humans. So that is very interesting to me, because so there you can think, okay, what would this add? A clinical pharmacologist, clinical pharmacology in general, that's actually what this talk will be about. What do we know about pharmacology of cannabinoids and of terpenes? And yeah, we don't know everything. We don't know everything. It's limited. But we know quite a lot. But everything we know, or many things we know, it is not applied in product development. And that is so unfortunate, because we could be so many more steps ahead if we would only apply what we know from research now or research before I was born in the 70s. It is out there, but we need to apply it. And we can apply it, and then products will be so much better. Anyway, I will give a short overview now of pharmacology and cannabinoids and terpenes. And I don't have a pointer, my slides are full. I'm sorry. Here we go. We start with cannabinoid pharmacology, pharmacokinetics, actually, of cannabinoids. So there are a few characteristics that I want to go over with you. Firstly, we know of the products out there that there are very various and also pretty unconventional administration forms of cannabinoid products. Unconventional because it's not like medication, but even one food product, you don't have that in 6 different administration forms typically. So that leads, depending on the product, of course, to sometimes even no absorption, very difficult absorption, or very variable absorption. Even if you have a very good quality product, a very well-known characteristic of cannabinoids is that if you take it or your neighbor takes it, it might turn out very differently. So that is very important to keep in mind. Lipophilic, the compounds are very lipophilic. That means that you have some difficulties with distribution, and you might have seen sometimes maybe a concentration profile with this lingering tail, you know, that can very well be, especially after, after different administration forms, it can very well be redistribution. So a compound is distributed to a tissue, and then at some point it goes back into the bloodstream, and that is something you see and you need to really take into consideration. High first pass metabolism, something else. It just came up at the previous session. So that means that, I'll just repeat because it's not so easy, such an easy matter, that if you take a compound as an edible, it will not directly go into your bloodstream to be distributed. It will first go into your liver. In your liver, large part of the cannabinoids will be metabolized. And some of those metabolites, as you also, that was discussed during the CYP29C, C9 discussion, will be transformed to, a form that is also active. But it's active in a different way than the parent compound is active. And if you bypass that by, for example, inhaling and bringing it directly into the systemic circulation, without it being broken down first, you can already see that that will lead to different effect profiles. Furthermore, multiple active compounds that can lead to pharmacokinetic drug-drug interactions. At the level of absorption, you get interaction. At the level of metabolism, you get interaction. So the pharmacokinetics of cannabinoids are pretty complex, and it has consequences for pharmacodynamics as well. I'm skipping that part. I'm going directly to some pharmacodynamic characteristics. There are two receptor subtypes. The endogenous cannabinoid system has two receptor subtypes. Cannabinoid receptor subtype 1 and 2, and that's it. If you look at different physiological systems, like today we heard the words of dopamine system and serotonin system, those systems have a lot of receptor subtypes. So the challenge is for cannabinoids, if you want, you cannot just say, I want to target that effect and that organ, because you only have two receptor subtypes to choose from. There are some tricks, but we have no time for that. It sounds pretty paradoxic in that sense, that the next point is saying this is not a single target drug. If you think, okay, with a plant cannabinoid, I'm now going to target the CB1 receptor. Well, good luck with that, because you will also target other physiological systems, receptors, ion channels, or transporters of other systems. So you will also get a lot of off-target effects, let's say. And also here, we are dealing with multiple active compounds. And that means that also pharmacodynamically, you will get interactions. For example, effects that enhance each other or level out, or you have competition at the receptor level. So the pharmacokinetics and pharmacodynamics of cannabinoids are pretty challenging for drug development, but this also gives opportunities if you understand what you're doing or where you want to go. So yeah, I now go on from cannabinoids to terpenoids, because, terpenoids, everyone's talking about it, everyone has ideas about it, potential this and this and this effect, but what do we actually know and what don't we know? So I summarized it here. We know from various cannabinoids or terpenoids that they bind to various receptors. From others, we don't know it. And what we also don't know is, how do these terpenoids actually metabolize? Because as we saw from cannabinoids, that can actually have serious effects on what effects you're expecting. And also, we don't really know what the particular cannabinoid terpene mixtures do. Furthermore, we know that terpenoids have various effects in animals. If you get into a dispensary, you see these nice posters with all these difficult terpene names and all the colors of the rainbow, and then what potential effects they give. Well, that's really nice. Those overviews were mainly made by observing one particular change in an animal. It is not translatable, because we are not only talking about different organisms that you need to translate to, we are also talking about different dosages, different administration forms. You don't administer your terpene in your belly with an injection. And all of this is very important with realizing that if we see something there, it's not necessarily translatable to humans. So we just don't know. What we do know is some effects of compound mixtures in humans, but mind you, all these studies were mainly done with terpene mixtures from sage, from rosemary, and I think you get it. That's not the same terpene mixture as we have from cannabinoids. I really don't want to make you depressive, but I just want to show you what we know, because we know things as well. And then if we want to look at isolates, because maybe you can put some isolates together and then make a terpene or a mixture that is comparable to cannabis, well, some isolates, there's actually no real studies with isolates in humans. I'm sorry. But we do have some studies where there is one more dominant, there we go again, more dominant terpene in a terpene mixture that gave effect. So we know that, and we know something about the effects there. It's just very difficult to translate the dosing and administration from there. Not everything is replicable, and especially it's not translatable necessarily to the terpene levels that we find in cannabis plants. Okay. Congratulations, Linda. You've now given an overview of pharmacology for cannabinoids and terpenes. What do they do in humans? Because everyone always wants to know, what effect does ABC give in a human? Well, I made a little overview on the next slide about that. So we first look at effects of isolated compounds, cannabinoids in humans. This is what we know. THC gives various effects in humans. And the ones that are actually registered at the FDA for some compounds are here. Those are chronic pain relief, anti-nausea, and anti-vomiting with a cannabinoid analog, THC analog, and synthetic THC. CBD, we also know that it can have some anxiolytic effect, so anti-anxiety effect, and also anti-convulsive effect. As you might have heard that last year, a CBD isolate was registered for this purpose. And then CBN, another one, you hear that CBN is the sedative cannabinoid. Well, If you look at CBN studies with a little bit of a substantial sample size from the 1970s and 80s, there's just no effect. So, no, not as an isolate. And then you have this whole list of other cannabinoids that people speculate what they might do. But they have not been studied in humans as isolates, so we don't know. About the terpenoids. Here, we've got limonene. Well, that is one of those compounds that was tested on a ward with depressive patients, and is potentially antidepressant. It was the dominant compound in a mixture. Linalool, same story, dominant compound in a mixture in a series of European studies with patients with anxiety disorders, potentially anxiolytic. All the other major terpenoids from cannabis, we don't know. We don't know as isolates. It's all speculation. So this is an overview of the individual compounds. Then there is something else you hear a lot about, and that has to do with compound mixtures. The entourage effect. What is that about? So entourage effect, what people mean by that term is that if you have a compound mixture, that should give a larger effect than when you have the sum of the individual effects. That is the entourage effect. Does something like that exist? Well, it became very popular after a study by Carlini in 1974 that saw that if you give an extract to patients, they were healthy volunteers, they will feel 4 times as high than THC alone. But that's about where it was left at. Because if you look at animal and in vitro data, you can find this entourage effect. But if you look at studies in healthy volunteers or patients, there's one patient study with MS patients that describes that there are more side effects, so not really the site you wanted to go to. But there's not clear evidence other than that. So there are studies that say there is an entourage effect. There are other studies, actually more human studies, that say we did not find this entourage effect. And then what about the combination of THC and CBD? For example, in Sativex, that's not registered in the United States, but in more than 30 countries around the world, it is. And from THC, CBD, we also know a little bit, but it's very complex to really understand what this does. Because, for example, THC induces euphoria, induces anxiety, and CBD is able to decrease that a little bit. But if you give a higher CBD dose and a smaller THC dose, then CBD can actually increase and prolong the THC effect. So everything is very dependent on dose and ratio, etc., etc. So it's not really straightforward. So we don't know too much about compound combinations, but we know a little bit. And we also have some knowledge about single compounds. But this knowledge is typically not applied in product development. So let's go quickly through some product examples. You don't need to be a pharmacologist to look at this one. I found this, I just browse online sometimes and look at products. So here we have a product that I found on some website, and it has CBD and was supposed to lower blood sugar levels. So let's play the pharmacologist with me, and let's look at the ingredient list and see how much sense this makes. That would be wonderful, right? Yeah, so, okay, but there are also some examples that are less straightforward. For example, this product, it's hugely popular in Colorado. So this is a CBD plus THC plus CBD vaginal suppository. And we look at the ingredients. It has cocoa butter. That means that it dissolves at body temperature. And then, there's something funny with the layout, but it has a dose of 60 milligrams THC and 10 milligrams of CBD. 10 milligrams of CBD is considered in Colorado as a single dose. And actually, the regulators think that's on the high side. So 60 milligrams of THC per dose is very high. Okay, let's have a look at this compound. Typically with little or no psychoactive effects. Interesting. So what do they use this for? Specifically formulated for relief from menstrual and pelvic discomfort. Okay. If I have menstrual cramps and I'm at home, it's silent. I just lay down for 5 minutes and continue with whatever I was doing. Yeah, that's my strategy. I lay down for 5 minutes and I continue. Okay, so how does this product work? Let's look at the slide. For enhanced results. Okay. Interesting. So that's how it works. So wait, it says until fully absorbed. Now I go back down, up, typically with little no side. Okay, so you have 60 milligrams of THC that gets fully absorbed. You have little or no psychoactive effect. How much pharmacological sense does this make? Okay, open-ended question. I will continue. So. What is really interesting to me as a pharmacologist is that these products can exist and that they are very popular. We can do so much better. We can do so much better. And I think if you make a product that actually works, can you imagine how many other products you can actually knock off? And I'm not saying that because I'm very competitive. I'm saying this because people pay so much money for products that don't work. And that is pretty frustrating for me. And on my online data gathering platform, I see that there is a huge discrepancy between what patients report what they want, what I see that they need, versus what product companies offer. So it is very important to get effective alternatives. How to get these alternatives. I will not go too much into detail, but I will give a few rough points of simple modifications and more complex science that is needed to bring you ahead of the curve. And one thing is, for example, changing ingredients. Of course, it can change your product's effects because of interactions or other beneficial effects. Each effect requires a different delivery mechanism. Very important because, sometimes you want an effect to last longer or to start earlier, and you have to make trade-offs there, but you really have to understand what effect fits with what administration method. And then if you think, I have something that really works well, I've got this stuff, you can inhale it, it works really well, I want to make it into an edible, that's fine, but you cannot just take the same stuff, put it in a capsule, and then think, now I've got an edible, because you've seen that there are many reasons why this can actually not lead to a good product. And furthermore, always account for inter-individual variability and stratification, and make that actually a very strong point of yours, because if you do stratification, you modify a product just a little bit and you can target so many more people. Stratification is very important. That's the wrong button. And something else that was brought up by the previous speaker is test your efficacy. And I know that not everyone can afford a randomized placebo-controlled clinical trial, and good luck with the regulatory agencies. But observational studies, post-market research, it was already mentioned, and I mention it again, it's very important. So if you want to be the strongest long-term player, I think it is very, very, very important to understand that you can distinguish yourself by including pharmacology, really straightforward things we already know and more complex things that we already know. So thank you very much, organizers, my team at Viridian Science, Canify. If any questions, just email me. I will leave it up to the moderator, but there is still time for a question, if anyone has questions. And otherwise, just send me an email.
I'm going to go ahead and kickstart it then. So, yeah, you mentioned it is challenging. Can everyone hear me? No? No? Okay.
Okay, let's go here.
I'll start up the discussion. So you had met, well, thank you. That was amazing. So one thing that you had pointed out at the end is, yeah, you make a good point. It's very hard to afford a randomized controlled clinical trial. I'm wondering, what about, so physiologically based pharmacokinetic modeling? Are we seeing more of that? So that might help bridge the gap between maybe some preclinical studies, some clinical studies that we have, some in vitro testing, and how has that been developed in the cannabis space?
Yeah, so pharmacokinetic models, I've actually worked on them myself in the mid-noughties, and I have not seen a lot of development there, unfortunately. I'm doing consulting work for one pharmaceutical company that is very interested in applying that, so that's very encouraging. I think that a lot of people who work in product companies in the cannabis space might not be so familiar with methodologies that are applied by pharma, and therefore might not even be familiar with this type of modeling. So I have not seen it a lot myself, unfortunately, because it's indeed a relatively cheap tool to make predictive modeling. Yeah. Yeah? I see someone else.
Thank you for your presentation. I wonder, is any people, they do some nanoparticle formulation for the patch or better delivery?
I find it difficult to hear you. Can you try to speak up?
Oh, sorry.
No.
I catch a little cold. Is any formulation like nanoparticles for better delivery for pharmacokinetic study? Do you know any people in the cannabis industry doing that?
Did anyone hear that and can tell that to me? Because I could not understand it.
Okay, sorry.
What delivery methodologies you see that are being successful, nanoparticles, liposomes, what's your assessment of
What delivery mechanisms we see that are successful?
Yeah, then nanoparticle delivery for pharmacokinetic study.
nanoparticles?
Oh, nanoparticles. Oh, yeah. Okay, so there, nanoparticles is a
Sorry,
whole...
I'm a little cold, the sound.
Yeah, sorry. Yeah, so nanoparticles is a whole different field. Nanoparticles is not one thing. Huh? You have so many different types of nanoparticles, and every nanoparticle has their own characteristic, their own need. There have been studies done with nanoparticles in animals mainly. There have been various claims, like you've seen really things that are almost too good to be true. So I would love to see someone replicating testing that. That's what you typically do to see if it's really working that well. And we yet have to see how this works out in humans, but it has potential in various ways for increased bioavailability, so how much actually enters your body, for prolonged release. So you take something and hopefully it will stay in your blood for a few days if you want that. So there are different things, applications possible. I see my time is done now.
Thank you.
But yeah.
One more short question.
Okay.
Okay, well, I have a short one, and I have a difficult one. I'll give you the short one first.
Okay, speak in the microphone.
Yeah, I'll speak up when I'm speaking into the microphone. Hi. Something I've always wondered, this mostly has to do with CBD, but it's a general question, and I've heard it referred to as being an inverse agonist, which has always driven me absolutely nuts. Exactly what the heck that is.
You're asking what an inverse agonist is?
Yes, is it a term that's even being used appropriately? In
Yeah.
your opinion?
If the question is what an inverse agonist
As opposed
is?
to being an antagonist to the CB1 or something like that.
Yeah, I find it hard to hear the microphone people.
I keep stepping away
But-
from the mic, I'm sorry.
It's, let's say a full agonist gives 100% effect. An antagonist gives 0% effect.
Okay.
An inverse agonist gives minus 100% effect. So it's actually what the opposite is of an agonist. I hope that helps.
Sounds like it's a little bit over the top, but that sounds good. I like that. The other question, and this is a little bit more, you mentioned about lack of PK on drug-drug interactions, and I was wondering if you were familiar with some of the CBD interactions where they were showing. Basically, my thing is high blood pressure medications and other medications that get whacked by CBD. Basically.
Yes, you're asking about the metabolic interaction of CBD with particular blood medications, right?
In a way, just with any medications that people like me are taking. It's changing the clearance, it's changing the renal clearance.
I didn't hear that last part. I'm sorry.
I believe, if I'm using the terminology correctly, it's changing the renal clearance. Okay, how long it sticks around.
I can't hear
By
it.
changing the inhibition. Okay, well, I guess that's about all I can say about it.
I can say one thing about blood thinner interaction, for example, because that is very. You have different types of medications. If you have a headache, you take medication, and your dose will go a little bit up or down. Yeah, that's just very unfortunate. You can have more headache, right? But if you have, for example, a blood thinner, you cannot just go a little bit more up or down because if your blood is too thin, you just die. And therefore, and these blood thinners, they work also via the CYP enzyme, the P450 enzyme system, like warfarin, for example. And if you have interactions with that enzyme system and cannabinoids, so therefore you have to be really, really careful and always ask your physician if you use those type of medicines and you are considering cannabinoids.
They don't know anything about cannabinoids, but thank you.
Thank you.